Usually when we talk about a medical diagnosis or just looking at the human body in general, there's this expectation of absolute precision, like engineering.
Right. Yeah, like a car engine or something.
Exactly. Like you break your arm, the x-ray shows that jagged white line on the radius, and the doctor just points at the screen and says, well, there it is.
Yeah, it's very binary. I mean, the bone is whole or the bone is broken.
Right. It's clean.
Yeah.
And I think we find a lot of comfort in that. We really like things to be visible, to be easily categorized into normal and abnormal. But then you step into the world of neurodevelopment and suddenly...
Oh, the x-ray machine is completely useless.
Completely useless. We're looking at a landscape that is just incredibly murky. I mean, think about a newborn baby. If you've ever spent time around one, they're basically just adorable, flailing chaos.
Yeah, they really are.
They can barely find their own mouth with their thumb. And yet, if you fast forward just a few years, that exact same human nervous system can perform these highly coordinated microscopic movements.
Which is wild when you really think about it.
Right. We're talking typing 90 words a minute on a keyboard, or threading a tiny needle, or playing a Chopin waltz on the piano.
It is the absolute definition of a biological miracle. But it's a miracle that's grounded in intense physical rewiring of the body.
And to figure out how we actually get that incredible coordination, today's deep dive is venturing into some truly groundbreaking territory. We are pulling from a recent Nature Neuroscience paper by Merovich and colleagues.
Yeah, this paper is just incredible.
It really is. It's titled Connectomic Evidence That Ordered Activity Drives Neuromuscular Network Formation. And we have all their research data, the imaging, the computer simulations, the works.
The whole package.
So our mission for this deep dive is to figure out exactly how physical experience and activity quite literally sculpt the muscles and nerves in your body.
And we really should clarify just how monumental the source material here is because, well, this isn't your standard biology paper. The researchers mapped complete connectomes.
Right. And a connectome, just to define it, is a comprehensive three-dimensional wiring diagram.
Exactly. But in this case, they mapped every single motor neuron and every single muscle fiber in a specific mouse muscle.
Every single one.
Yeah, the interscutularis, which is a tiny ear muscle. And they tracked its development from the exact moment of birth all the way to adulthood.
Okay, let's unpack this for a second. Why an ear muscle?
Well, mostly because of its size. I mean, to map an entire connectome at a cellular level is incredibly computationally heavy.
Oh, what a bet.
So by choosing a small, self-contained muscle, they weren't just looking at a tiny representative sample of a few synapses, which is, what we usually do in these studies.
Right. Usually it's just a snapshot.
Exactly. But here, they were able to look at the exact physical fate of every single neural connection in that entire muscle over a lifespan.
Which means to understand how we get so perfectly coordinated, we first have to look at the absolute chaos of how we are wired on day zero, the starting line.
Right. So the researchers looked at the P0 connectome P0, meaning postnatal day zero, so literally the day the mouse is born.
Fresh out of the womb.
Yep. And to actually map this, they used a technique called serial section electron microscopy, or SEM.
Okay, break that down for me. What did that actually look like in a lab?
Okay, if you want to visualize this, imagine taking this tiny ear muscle, freezing it solid, and then using a diamond-edged knife to slice it into layers.
A diamond knife.
Yeah, because they need to slice it thinner than a single wavelength of light.
That is absurdly thin. I can't even picture that.
It is. So you take a highly detailed electron microscope picture of every single one of those thousands of slices, and then a supercomputer basically stacks them all back together into a navigable 3D model.
So they are literally looking at the cellular wiring at a nanometer scale, just wandering through the muscle like a video game environment.
That's exactly what it's like. And in this tiny newborn muscle, they tracked exactly 20 motor axons.
And axons are the long cables coming out from the nerves in the spinal cord, right?
Right, the cables. So 20 axons and 240 individual muscle fibers. And what they found at the moment of birth was absolute, overwhelming redundancy.
What do you mean by redundancy?
Well, every single muscle fiber receives input from multiple different axons. In fact, some individual muscle fibers were being innervated or plugged into by 75% of all the motor neurons in the entire muscle.
Wait, 75%?
Yeah, it's massive.
Okay, let me do a quick analogy here. Imagine a single worker on a factory assembly line. That worker is the muscle fiber. Now imagine that worker is receiving simultaneous conflicting instructions from 15 different managers screaming through multiple megaphones all at once.
Yes, that is a perfect way to look at it.
That's a newborn's muscle fiber, just pure noise.
It's incredibly noisy, and to understand just how noisy, we have to look at the physical connection point itself.
Right.
So the junction between a nerve and a muscle is called a neuromuscular junction, or NMJ.
NMJ, got it.
Now, in a typical adult, one muscle fiber generally has exactly one NMJ. It's a very clean one-to-one relationship.
It's one boss for one worker.
Exactly. But at birth, the researchers found that 34.4% of newborn muscle fibers have more than one NMJ.
So over a third of the fibers are double wired.
Or triple wired.
Oh, wow.
Yeah, we call these MEFs, or multiple end-plate muscle fibers. Some of them had 3 completely separate junctions physically located on the same muscle fiber.
That sounds like a mess.
It gets worse. At each of those individual junctions, there could be up to 13 different axons all crowding in, trying to connect to the exact same spot.
13. Okay, so before we move on, let's just do a really quick recap of where we are. We're at day 0. The muscle is an absolute tangled web of overlapping electrical signals.
Total chaos.
If I'm that factory worker getting yelled at by all those bosses at 3 different workstations, I'm quitting. Or, biologically speaking, the body needs to somehow fire most of those bosses to get any actual work done.
Right, it has to clean house.
So if you're listening to this and wondering how on earth a baby ever manages to move with purpose, you're not alone. So how does the muscle actually go from that chaotic P0 state to the streamlined adult state?
Well, to see that, we have to jump forward in the data to postnatal day 60, or P60, which is the adult state for a mouse, to see what actually changed.
Okay, day 60.
This is the period of what we call the great pruning. By adulthood, that incredibly messy system is heavily streamlined. The number of axons innervating each NMJ decreases about 6.5-fold.
So all that crowding at the actual junction, all 13 bosses fighting for the megaphone, that just goes away.
Completely. Every single neuromuscular junction in the adult mouse only has one single axon attached to it. It goes from 13 down to 1.
Wow.
And those MEFs, the muscle fibers with multiple junctions scattered along them.
The ones that were about 35% at birth.
Right. They drop drastically down to just 6% in the adult.
That is a massive teardown.
Yeah.
From 35% having multiple connections down to just 6%?
It's ruthless. And the paper discovered there are actually 2 concurrent forms of synapse elimination happening to achieve this cleanup.
Okay, what's the first one?
First, there is the well-documented local competition. This is what's happening when multiple axons are crowding a single junction, fighting for territory until only one wins and the others retract.
So that's the 13 bosses fighting at one desk until one wins.
Exactly. But the researchers also discovered a completely new long-distance competition.
Long distance, what does that mean?
This is a mechanism where entire NMJs, like the whole junction itself, the desk in your analogy, are completely destroyed and eliminated from the muscle fiber.
Wait, an entire junction just wiped off the map?
Yes, gone. And crucially, this long-distance elimination is entirely dictated by physical distance.
How so?
When researchers looked at that rare 6% of surviving MEFs in adults, those few muscle fibers that actually managed to keep 2 junctions, they noticed a pattern. Those 2 surviving junctions were always extremely far apart from each other. If 2 junctions are too close together, meaning they're separated by less than 50% of the muscle's maximum length, one of them is always destroyed during development.
Wait a second, I have to push back here.
Sure.
How does a neuromuscular junction know it's too close to another junction on the very same muscle fiber? I mean, they don't have eyes. They can't look down the length of the fiber with a tiny measuring tape.
Right, they have no spatial awareness.
So how are they fighting each other from afar?
That brings us to what I honestly think is the most fascinating mechanical explanation in the entire paper. They call it the action potential collision theory.
Action potential collision. Okay. To understand this, we probably need to picture how a muscle actually receives a signal from the brain in the first place.
Exactly. So when a nerve fires, it releases a chemical neurotransmitter right at the NMJ.
Right.
But that chemical signal immediately triggers an electrical signal on the muscle fiber itself. This electrical wave is called an action potential, or AP. And this AP physically travels along the outer membrane of the muscle fiber, sweeping down its length to make the whole thing contract.
So it's like a wave of electricity moving down the muscle, kind of like dropping a stone in a pond and watching the ripple travel outward.
That's a great analogy. A very fast ripple, yes. Now, think about what happens if a single muscle fiber has two NMJs on it, and they fire asynchronously.
Asynchronously meaning the brain sends a signal to them at slightly different times.
Exactly. Even just a tiny fraction of a millisecond apart.
Okay, I'm visualizing the pond. The first junction fires, dropping its stone. It sends that electrical wave down the fiber.
Right. And because the second junction hasn't fired yet, its structural doors are effectively open. It's just sitting there waiting for a signal.
Okay.
So that electrical wave from the first junction physically invades the second junction.
I'm assuming biological invasion is a bad thing.
Very bad. That incoming wave destabilizes the chemical receptors at the second junction. It scrambles the hardware.
Yikes.
And if that happens enough times, the second junction is fundamentally damaged and eventually destroyed by the muscle.
Wow.
This is the mechanism for the long-distance competition. The first junction to fire sends an electrical assassin down the fiber to wipe out the competitor.
An electrical assassin. That is incredibly metal. But wait, if asynchronous firing sends an assassin, how does any fiber ever survive with two junctions? I mean, how do we get that 6% of adult MEFs that somehow made it through the pruning process?
Well, they survive through synchronous firing.
Firing it at the exact same time.
Yes. If both junctions receive a signal from their respective nerves at the exact same time, they both generate an action potential simultaneously.
Okay, so two stones dropped in the pond at once.
Right. And those two electrical waves travel toward each other and crash right in the middle of the muscle fiber.
And they cancel each other out?
They literally annihilate each other.
Yeah.
Because they collide in the middle, neither electrical wave ever reaches the other junction. The physical collision protects them both from invasion.
Okay, let me try another analogy here. Think of it like two trains on the same train track, and the trains represent our electrical signals.
Okay, I follow.
If both trains leave their stations at the exact same time, they meet in the middle of the track and crash. The train stations, which are our neuromuscular junctions, are perfectly safe.
Right.
But if one train leaves early, which is our asynchronous firing, it barrels all the way down the track and completely destroys the other station.
That train track analogy is spot on, and it perfectly explains why distance is the deciding factor for survival.
Because of the travel time.
Exactly. The further apart the two junctions are, the more time there is for a collision to happen before one signal can cross the entire distance to reach the other side.
Right, because if they are too close together, even a tiny microsecond delay means the train hits the station before the other train can even depart.
Precisely. And the researchers actually mapped out the exact math on this.
Oh, really?
Yeah, they found that the electrical signal travels along the muscle at a speed of about 1.5 meters per second.
Which is pretty fast.
It is, but there's also a tiny random biological delay at the synapse itself of about 0.5 milliseconds.
Just natural biological lag.
Right, so even if the brain perfectly coordinates the signal, the actual muscle firing might be slightly staggered.
Ah, I see.
So they calculated that to guarantee a 95% collision rate, basically, to guarantee that those train stations survive, the two junctions must be separated by at least 790 micrometers.
Wow. Okay, time for another quick summary check. So for a double connection to survive the great pruning, it all comes down to two things: synchronous firing and physical distance.
You nailed it.
But that raises a massive question. If survival requires firing at the exact same time, how does the brain actually coordinate that timing?
That's the million-dollar question.
Right, like how do different nerves manage to fire synchronously enough to save their connections?
Well, to figure that out, we have to look at the rules governing when neurons fire. And that introduces a foundational rule of neuroscience called Henneman's size principle.
Okay, Henneman's size principle. But before we get to the rules, let's just clarify some terminology so we don't get lost.
Good idea.
The paper talks a lot about motor units here. Is a motor unit just a nerve?
Ah, good catch. No, a motor unit is a package deal. It is one single motor neuron coming from the spinal cord, plus all the individual muscle fibers that it plugs into.
Got it. The boss and all their specific workers?
Exactly. So Henneman's size principle.
Right. What does size have to do with firing order?
Well, when your brain wants to contract a muscle, it doesn't just turn on all the motor units at once. It recruits them in a strict fixed order based on size, starting from the smallest and moving to the largest.
Why start small?
Because small motor units, meaning a small neuron, controlling just a few small muscle fibers, they produce a very weak contractile force, but they are highly fatigue resistant.
Oh, so you use them constantly for things like maintaining your posture or picking up a feather.
Right, they are the slow, steady workers. You don't need maximum power to hold your head up.
Makes sense.
But if you need more force, say, you're suddenly lifting a heavy dumbbell, your brain then recruits the larger motor units on top of the small ones.
Like shifting gears.
Exactly. These large units connect to many massive muscle fibers. They produce huge force, but they get tired really quickly. But the golden rule here, the absolute rule, is strict. The small ones always fire first.
So the body always scales up the power as needed. It never starts with the big guns.
Never.
Okay, I understand the theory. But how did the researchers actually see this strict firing order playing out in a microscopic mouse muscle?
Oh, this is my favorite part. They used one of the most visually stunning techniques in modern biology. They used what are called brainbow mice.
Brainbow. Like a rainbow of brains. I love it.
It's an amazing transgenic technique. Basically, by inserting genes for red, green, and blue fluorescent proteins into the mouse genome, the neurons randomly mix these colors as they grow.
Like mixing paint.
Exactly like mixing paint. And as a result, every single motor neuron glows in a distinct, unique fluorescent hue under the microscope.
Oh, wow. So you can quite literally see the wiring diagram in Technicolor.
You really can.
The images in the paper are incredible, by the way. It looks like a neon art installation inside the muscle tissue.
It really does. So they looked at these brainbow muscles between postnatal days 6 and 9, which is right in the middle of this intense pruning process we talked about.
A great pruning.
Right. And they found a strict rank-ordered connectivity at the junctions.
What does rank-ordered mean in this context?
Well, for example, a red neuron shared dozens of junctions with the blue neuron right next to it in the recruitment sequence.
Okay.
But that same red neuron shared almost 0 junctions with the green or magenta neurons that fire much later in the heavy lifting sequence.
Ah. So neurons of a similar firing time stick together.
Because they fire at roughly the same time, their electrical waves collide, protecting their shared junctions. The nerves that fired synchronously survived together. The ones that fired asynchronously invaded and destroyed each other.
That is just wow.
I know. The wiring diagram of an adult muscle is quite literally a physical fossil record of the electrical activity that happened when it was developing.
It's a map of its own history.
Exactly.
But here's where it gets really interesting, though, because as I was reading, I noticed a really cool quirk in the paper about those small, early recruited nerves. It's a phenomenon they call motor unit rotation.
Yes, this is a brilliant mechanical twist.
So you'd think the smallest units, since they are used constantly for posture and light movements, would fire together all the time and share a ton of junctions, right?
Well, that would be the logical assumption, based on the size principle.
Right. But to prevent fatigue, those small, early recruited tonic motor units will actually alternate their firing. One rests while the other works, and then they switch.
And if they are taking turns...
That means their signals are technically asynchronous.
Yes.
They are intentionally never firing at the exact same time. And according to the action potential collision theory, asynchronous firing means invasion.
It means destruction.
So because they take turns, they are actively sending electrical assassins to destroy each other's connections.
Which perfectly explains why they actively avoid sharing the same junctions in the adult muscle.
It is a stunning display of form following function. The physical architecture of the muscle is ruthlessly pruned into peak efficiency based entirely on how it is used.
It's breathtaking. Biology acting as its own engineer.
It really is. But that leaves me with one lingering question.
Lay it on me.
Okay, so we know action potentials colliding protects nerves. We know asynchronous firing prunes them. And we know this pruning is strictly guided by the size principle. You know, when you are recruited, it dictates who you survive with.
Oh, correct.
But when does this all actually start? I mean, does this competitive pruning just magically turn on the second the mouse is born and starts wiggling around in the nest?
That was a mystery the researchers wanted to solve too, because remember earlier, we established that the newborn P0 connectome was completely chaotic with massive redundancy.
Right, the 15 screaming bosses.
Right, so they ran advanced computer simulations to see if you could just apply random pruning to that messy P0 connectome and eventually arrive at the neat, organized adult connectome.
Let me guess. The simulations failed.
Completely failed. You cannot get the highly specific adult state through random synapse elimination.
So that implies there has to be some kind of underlying structure or bias already present on day 0.
Exactly. The deck is already stacked. When they looked incredibly closely at the P0 newborn data, they found a faint but definite structure already there.
Like what?
While even at birth, the largest motor units are already preferentially innervating the largest muscle fibers. It isn't 100% random chaos.
But wait, if the baby mouse hasn't been running around yet, how is activity-driven pruning already happening before it's even born?
And this raises a profoundly important point about development. It strongly implies that activity-driven pruning doesn't just start when the animal is born. It is highly likely that prenatal motor activity kicks off this massive neural organization.
Prenatal activity. So if you're listening to this and wondering what this means for human development, I mean, think about a pregnancy.
Oh, it changes how you look at the whole process.
It means those little kicks and flutters a pregnant mother feels in the 2nd and 3rd trimester aren't just the baby stretching its legs or, getting cramped in there.
No, not at all.
That is the baby's nervous system running real-time hardware updates. It is actively firing those nerves, forcing action potentials to collide, intentionally destroying the weak connections so it can build a customized, gapeless, ultra-efficient neural network before it even enters the outside world.
The nervous system is actively testing and sculpting its own hardware based on the physical feedback of the body in the womb.
Yeah, it's just unbelievable.
It really is a marvel.
So we started this deep dive with a pretty basic question. How do we go from a clumsy, flailing newborn to a highly coordinated adult capable of playing the piano? We looked at the connectome of a muscle from birth to 60 days. We saw that we start with a chaotic web of overlapping nerves, but through the literal physical collision of electrical signals, those action potential collisions.
Right, the trains crashing on the tracks. And guided by a strict firing order based on size, our bodies prune away all the biological noise. We eliminate the redundant nerves, we wipe out entire junctions that are too close, and we leave behind a perfectly efficient system.
The great pruning.
Yeah. This isn't just biology, this is the literal blueprint of how we gain physical coordination.
And this paper proves that our neural circuits are a physical imprint of our past movements.
A fossil record, like you said.
Exactly. Which raises an important final question for you to mull over.
Oh, I love a good lingering thought. What is it?
Well, in neuroscience, this intense pruning window in childhood is largely considered closed by the time we reach adulthood.
Right.
The hardware is set.
Yeah, but if someone undergoes massive physical rehabilitation after a traumatic injury, or say they dedicate themselves to learning an entirely new complex physical skill later in life, like mastering a martial art or the violin, are we merely running new software on old fixed hardware? Or might there be a hidden mechanism that actually reopens this competitive pruning process, allowing the body to physically rewire its connectome once again?
Oh, man. A fascinating thought to leave on. Maybe that adult brain is still capable of rebuilding the train stations.
It's definitely something to think about.
Well, thank you for joining us on this deep dive into the architecture of movement. Keep questioning, keep exploring, and we'll catch you next time.